Hello, BISC1323. This is the beginning of recorded lecture 2.5. We briefly began this chapter in the last one, and now we're going to continue on talking about plant form and physiology. So we talked about one of the four types of plant tissue in the previous lecture, but now we'll do the other three. So starting with dermal tissue. If you're familiar with the word dermal at all, referring to your own body, dermal means skin, and it's the same way with plants. Dermal tissue is their outermost layer. Here's the D for dermal tissue. So yeah, there's dermal tissue on the leaves, there's dermal tissue on the stems, there's dermal tissue on the roots. It's the outermost layer of cells everywhere on the plant. It covers and protects the plant. In all parts of the plant and in all plants, it forms what's called the epidermis. Key terms defines the epidermis as a single layer of cells found in plant dermal tissue, covers and protects underlying tissue. Pretty straightforward. So they all have epidermis, but some plants also have bark, which is also in the key terms, a tough, waterproof outer epidermal layer of cork cells. So, here we know what bark is, but it refers to all these layers of things outside of the inner tissue. Now, plants need to breathe. So this is kind of a weird notion. We don't think of plants breathing, especially not with lungs and stuff like we breathe, but they do need to perform gas exchange. They do need to acquire CO2 from the surrounding air so they can fix it into glucose through photosynthesis, and they need to get rid of O2, a byproduct of photosynthesis. So in their leaves, they need openings. Again, the dermal tissue is great at covering everything, but it can't cover everything. They need openings, and these are called the stomata. They allow for gas exchange. Now, these openings are required, but they're also kind of a liability. You don't want too much stuff going in and out of the plant tissue. So there are specialized cells called guard cells. Sorry, I've been not showing the picture here. Yeah, these are the stomata, also known as stoma. I prefer the word stomata, but some of the figures say stoma. Here's the opening, and yes, these guard cells on either side of this are specialized cells of dermal tissue that can close and open in order to control how much evaporation, how much gas exchange is happening. So guard cells, specialized dermal tissue used to line these stomata and control their opening. There are a couple of other examples of specialized dermal tissue. Another one is something called trichomes. So these are fuzzy little hairy extensions. Their job is, not unlike our body hair. It helps provide a little bit of shade to the tissue underneath that. But also these trichomes can sometimes house toxic substances to discourage herbivores. So we see a variety of plants with these trichomes. In summary, hair-like structures, they can prevent water loss, again, providing some shade, and or store toxic compounds to discourage herbivores. And here's our next example of sort of specialized dermal tissue, root hairs. They're real straightforward. They increase root surface area for absorption. So here's a plant root coming out of the seed, and you can see the root hairs coming out of the root. Again, when you're absorbing stuff, you want as much surface area as possible, and they help increase surface area. Point of order, fungi are still better. Hyphae and mycelium is still better, but this is plants' attempt at trying to have as much surface area as possible. And again, specialized dermal tissue. So, root hairs increase surface area. Now, our third type of tissue in plants is vascular tissue. And hey, this is a familiar one. We already kind of mentioned vascular tissue. It was one of those evolutionary innovations that separated land plants into things that don't have vascular tissue, the bryophytes and the other groups that do. We can see vascular tissue on this figure. It's the V throughout the plant. And there's obviously vascular tissue in the stems, vascular tissue in the leaves, vascular tissue in the roots, located all throughout the plant. Now, what we didn't do last time I mentioned vascular tissue was break down the two different types. So these two types of vascular tissue are called xylem and phloem. Here's some xylem. Not much to look at. It's just tubes. Um, the wider tubes are called vessels, and the thinner tubes are called tracheids. Trachea is where these tracheophytes come from. Remember, that was another name for vascular plants. So the job of xylem is to transport water. Um, the walls of this xylem are made up of dead cells. Tracheids are narrow tubes, vessels or vessel elements are the wide tubes. But yeah, this is going to transport water from the roots, where the water is, up to the parts of the plant that need the water. The other type of vascular tissue is called phloem. It kind of looks the same. It's tubes in the plant, but it has some notable differences. The cells in the walls of the xylem were dead, but the cells in the walls of the phloem are actually living cells. It's really hard to stay alive in this sort of stretched out, elongated shape. And so there are companion cells nearby that help support the metabolism and life of these phloem cells. Now, this system of transport is not for moving water, it's for sugars, or sometimes I think of it as food, just to remember which one is which. Phloem is food, xylem is the other one. I can't think of a synonym for water that starts with X, but if you remember, is food and phloem, xylem's the other one. So these are transporting the products of photosynthesis to all the rest of the plant that needs those sugars. So phloem transports sugars. Living cells make up the walls. And we've got these sieve tube cells that make up these walls that are supported by what we call companion cells. Together, and there's an important reason why we're always going to find these together, we'll get to at the end of this chapter, xylem and phloem together make up what are called vascular bundles. And that's a phrase that we already saw at the end of the last chapter when we were talking about the differences between eudicots and monocots. And so, here are the vascular bundles, xylem and phloem, around the outside, and eudicots scattered throughout the stem in monocots. But the term vascular bundle just refers to both of these together. Okay, one more type of tissue in plants, and it's called ground tissue. It's the G that we see in this figure. And so ground doesn't refer to like above ground or underground because it's found everywhere. You'll find ground tissue in the roots. You'll find ground tissue in the stems and in the leaves and the tops of the shoot. You'll find this everywhere. Ground tissue is responsible for pretty much everything that we haven't covered already. So all other functions the plant would need are performed by ground tissue. So this includes photosynthesis, this includes storage of carbohydrates, and this includes just physical structural support of the plant itself. Now, there are 3 cell types of ground tissue. They're not much to look at visually, but here they are if you want something. Parenchyma, collenchyma, and sclerenchyma. Now, if we lived in a logical world, one of these would do photosynthesis, one would store carbohydrates, and one would do structural support, but that is not the case. I guess it's kind of the case. Let's read the key terms definition. So the definition of parenchyma is the most common type of plant cell, site of photosynthesis and starch storage. So parenchyma does these first 2 functions. Okay, what about the other 2? Collenchyma is, reading from the key terms, elongated plant cells with unevenly thickened walls, semicolon, provides structural support to stems and leaves. So parenchyma does photosynthesis and storage. Collenchyma provides structural support. Now you might be thinking, what is there possibly left to do? What about the third one? Well, sclerenchyma reads, plant cells with thick secondary walls to provide structural support, semicolon, usually dead at maturity. So collenchyma and sclerenchyma are both responsible for structural support, but these are living cells when they're mature. The sclerenchyma are dead cells when they are mature. So that's the 3 cell types and are all defined in the key terms by what function they provide as ground tissue in the plant. Now, we've broken the plant body down into root system and shoot system. We've broken it down by tissue type. There's one more way that we can break down and talk about the anatomy of a plant, and that's by talking about the major anatomical structures: stems, roots, and leaves, starting, as you can see, by talking about stems. So obviously, the stem is part of the shoot portion of the plant, and its job is pretty straightforward. The stems support the leaves, support the weight of leaves and flowers, if you got them, these reproductive structures. There are a couple of important key terms when it comes to talking about stems. Stems have nodes and internodes. Let's take a look at these. So the node is the point along this stem where a leaf is attached. I think the key term says, yeah, a point along the stem at which leaves, flowers, or aerial roots originate. More on aerial roots later, but it's where stuff comes off of the stem. The term internode simply refers to this space of the stem in between the nodes. So here's a node, this blank stem in between the two is called an internode. The key term says an internode is the region between nodes on the stem. So pretty straightforward terms, but we got to be familiar with these going forward. So the stem itself is pretty boring, has a pretty straightforward job. What is less boring, in my opinion, is when stems are modified to do other jobs. There are a lot of relevant modified stems. Let's go through these one at a time. So let's start with something called a rhizome. A rhizome, well, let's just take a look at some of these. So we see rhizomes in ginger, we see rhizomes in bamboo. It's labeled right here, rhizome. A couple of things are atypical about this. One, it's underground. Stems are typically above ground because they're supporting the weight of leaves, but a rhizome is indeed an underground stem. The other weird thing about rhizomes is that they go side to side. They run horizontally. Again, usually stems go up and down because they're supporting leaves above them. So these rhizomes, to read the key terms definition, a modified stem that grows horizontally underground. Why would you want to do this? Well, at the nodes here, new plants can originate, or new shoots can originate from these nodes. So yeah, bamboo is great at doing this. You may walk into a cluster of bamboo, a small bamboo forest or break, I think is what it's called, and it may look like there are 30 different bamboo plants growing out of the ground. In reality, it's probably just one single individual bamboo plant, but they've all connected themselves. It's one thing that's sort of spread out side to side underground. So this is a way for a plant to spread itself, putting up new shoots at these nodes. So again, underground, runs horizontally, new shoots can emerge at these nodes. Now, there's a similar thing called a stolon. A stolon is also a modified stem that runs horizontally. This figure is a little misleading because it makes it look like the stolon is above ground and the rhizome is below ground. That's not necessarily true. Some stolons are above ground, some stolons are below ground. The important difference between a stolon and a rhizome is the size of the internodes. Look at this rhizome here. You've got node, internode, node, internode, node, internode, node. These internodes are pretty short. But in a stolon, you have a node and then a long internode before you have another node. So again, very similar, but the distinction is important. The key terms define a stolon as a modified stem that grows horizontally underground or above ground. New shoots can emerge at the nodes, just like rhizomes, but there are longer internodes than rhizomes. That's the big difference. There's one more horizontally running stem, something called a runner. Strawberries make runners. If you've ever grown strawberries, they'll try to escape their bed. These are horizontally running stems. What's the difference between these and everything else? Well, honestly, sometimes they're used interchangeably with stolons. The key term says a runner is a modified stem that grows horizontally above the ground. New shoots can emerge at the nodes, like I've said with all of these, but again, sometimes it's used interchangeably with stolon. I'm not going to try to trick you on a test question. Rhizomes are underground. Stolons could be above ground or underground. Runners are above ground. And because these can both be above ground, sometimes they're used interchangeably. But all three of these run horizontally and can make new shoots at the nodes. Okay. Something different, another modified stem. Stems can also be modified to store carbohydrates. So that's what a tuber is. Here's probably the most famous tuber, the potato. Let's zoom in a little bit. Oh yeah, here are some stolons, below ground stolons, because remember stolons could be above ground or below ground. The tuber is really a structure that forms at the tip of this underground stolon. So let me read the key terms definition of a tuber. A modified underground stem adapted for starch storage. And so, yeah, this can be at the tip of a stolon, or it can be at the tip of a rhizome. So it's a modified rhizome or modified stolon whose job it is to store food. Similarly, there is a structure called a bulb. A bulb, again, we see plenty of these in our everyday life. We're talking about garlic, we're talking about onions, we're talking about tulips. Let me read the key terms definition of a bulb. It is a modified underground stem made of a large bud surrounded by numerous leaf scales. And yeah, if you've ever cut open an onion, you would see these layers, these scales. That's an important definition of what makes a bulb. And yeah, it stores carbohydrates. It stores food at the base of the stem, and importantly, it has layers. There's one more important modified stem that stores carbohydrates, and that is something called a corm. Not corn, C-O-R-M. A corm is probably the most notable example is this taro corm. To read the key terms definition, a corm is a modified underground stem that is rounded and fleshy. And yep, here's our corm. Yeah, this is poi. It's what taro is used to make a traditional Hawaiian dish. Yeah, this also stores carbohydrates at the base of the stem. Yep, stores carbohydrates at the base of the stem. What makes this different from a bulb is that it doesn't have any layers to it. You cut this open and it kind of looks like a potato, but again, don't get these confused. They all store carbohydrates, but the tuber is a modified rhizome or stolon. The bulb is located at the base of the stem and has layers. The corm is also located at the base of the stem, but it does not have layers. So again, all doing a similar job, but there are important distinctions between these three. Now, in all of this, you might be wondering why these aren't roots. You know, this is below ground. The onion is below ground. The potato is below ground. Well, the reason why these are modified stems is because they come from a part of the plant that is a stem. Even though they're underground, they're not doing the job of a root. There are actual roots. Yeah, here are some potato roots, and here are some roots coming off of the onion here. This structure is not a root because it's not doing the job of a root. It's not absorbing stuff from its surroundings. So the fact that it's underground doesn't inherently make it a root. It is a modified stem. Okay, two more, and these are easy ones. Tendrils and thorns. A tendril is a modified stem. I'm reading the key terms. A modified stem consisting of slender twining strands used for support and climbing. So it takes a lot of energy to be a big tall tree. You want to be a big tall tree because you get, closer to the sunlight, but it's much easier to get closer to the sunlight if you cheat and grow on other plants and, climb like that. Grapes do this, a lot of things do this. Ivy does this, cucumbers do this. Just wrapping around artificial or natural structures. That's a tendril. And yeah, thorns, I think we all know what thorns are. Let me read the definition. A modified stem branch appearing as a sharp outgrowth that protects the plant. And it's supposed to discourage herbivores. So these are also modified structures of the stem. All right, enough of stems. I said we were doing stems, roots, and leaves. Now let's move on to roots as an anatomical part of the plant. And obviously, roots are part of the root system. We already defined that. Just a quick callback, we already talked about different types of root systems when we were comparing and contrasting monocots and eudicots in the previous chapter. But what we can talk about here is by zooming in more closely at the root to look at the different zones that exist at the very tip of the root. So here we have the apical meristems. We already talked about these being the actively dividing cells, and they're near the tip of the root, but these apical meristems are not like at the tippy tip of the root. That would be dangerous. These are very important cells. We want them protected as plants. We want them protected because if these are damaged, if these are lost, it's very difficult for the plant to continue dividing. So at the very tip of the root is this root cap, which protects the meristems. And all of this is at the bottom zone, known as the zone of cell division, because that's what meristems do. They do cell division. So 3 zones at the root tip. Here's our zone of cell division, located at the bottom. This is where root meristems divide, and the tip is protected by this root cap. After that is the zone of elongation. In the zone of elongation, the cells that were produced down here elongate. They lengthen. There's no better way to put it. Not much else is going on here, but the cells are elongating. And in the middle, zone of elongation, cells lengthen. And then closest to the top is what's called the zone of differentiation. So when a cell is created by an apical meristem cell dividing itself, that cell is kind of a blank cell. It could be anything, but as it grows, as it matures, as it elongates, it's going to make some decisions about what it's going to be when it grows up. And these cells are going to differentiate into vascular tissue cells or ground tissue cells or dermal tissue cells and make these permanent decisions about what they're going to be at maturity. So at the top is this zone of differentiation where cells become specialized types. Now, just like we had modified stems, there are also modified roots. I promise you there aren't as many of these. So really there are just 2 categories of modified roots. One of them is starch storage roots. So just like there were several stems that stored food, there are also roots that can store starch for food. Carrots, parsnips, beets, rutabagas, all of these things. And again, the difference between these and the food storage stems is these are, because all of these things are underground, but these are roots. They do root jobs. They absorb stuff like a root does. These things don't. So these are food storage roots. These are food storage stems. The other type of modified root I want to point out are prop roots and aerial roots. So these are both roots that are above ground, which is weird because roots are typically below ground, but the prop roots are above ground and help brace these plants against wind or flooding. This is a corn plant we're looking at here. And aerial roots, as the name implies, are in the air. They never touch the ground. These roots wrap themselves around the branch of another plant and they absorb water from the air. and they absorb mineral nutrients from whatever rains down and sort of trickles down along the branch of this tree they're growing on. These are not parasites. They're not stealing nutrients from the plant they're growing on, the tree they're growing on, but yeah, their roots are, fully within the air. So I'm just saying that these are both types of roots that are above ground. Okay, stems, roots, leaves. Let's talk about leaves. So let's start by looking at leaf structure. So there are several anatomical parts to a typical leaf. Although pop quiz, is this a monocot plant or a eudicot plant? I'm not going to do the Dora the Explorer thing and make you call back to the computer, but yeah, you should look at this and know that this is a eudicot plant because the veins are sort of branching out. They're not running parallel. Anyway, let's start by talking about what's called the lamina. The lamina of the leaf is the surface. And it's protected. So if we zoom in on a cross section, we've got the cells that do photosynthesis. There's a stomata. There's some guard cells on either side. But if we look at this dermal tissue on the outside, there's this cuticle. So this is made of a layer of waxes that are going to protect this dermal tissue. So that's an important part of this lamina or this surface. The waxy cuticle helps it repel water like this. So the lamina is covered by a waxy cuticle. It protects the leaf. Another important part is the petiole. This is the part of the leaf that attaches it to the rest of the plant, to the stem. So petiole attaches leaf to stem. We also have vascular tissue. So we know what veins are. They're vascular bundles, and their branching pattern here. The midrib refers to the vein that runs the length of the leaf in a eudicot. Again, we only see this branching pattern here in eudicots. So veins are just the vascular tissue within this leaf. And yeah, just a reminder, monocots, eudicots, looks different. The midrib, as I said, is eudicot only. It's this major lateral vein from which other veins grow out. Now, all leaves have, these basic structures, but not all leaves look the same. There are many different types of leaf shape that we could talk about, but I just want to break down these two major categories of leaf, simple leaves and compound leaves. A simple leaf is a single undivided lamina, whereas a compound leaf, this is not 7 leaves here. This is a single leaf with a single petiole, but the lamina has been divided into, it's not always 7, but into multiple leaflets. So simple leaves have an undivided lamina. Compound leaves have a lamina divided into many leaflets attached to this midrib here in the center. Okay, just like we did for stems and roots, we talked about generic leaves. Now let's look at modified leaves. So there are several ways that we can modify a leaf to do different jobs. Let's start by talking about spines. So we find these in cacti, and they're kind of weird because they do the exact opposite job of what a leaf typically does. Normally, the job of the leaf is to have as much surface area as possible, to be flat and broad, because these leaves, they want to capture as much sunlight as possible to do photosynthesis. Doing photosynthesis is kind of their thing. But leaves are also a place where water evaporates, where water can be lost through these stomata. So in doing gas exchange, you're also going to lose water. In desert plants, losing water is a death sentence. And so instead of having leaves with as much surface area as possible, they have leaves with as little surface area as possible to minimize the amount of water loss. And instead, they're doing most of their photosynthesis through cells on the stem. Additionally, instead of just getting rid of their leaves, they're using them for the very important purpose of defending against herbivores. All these leaves have some sort of cuticle surrounding them, as we saw a few slides ago, but the cuticle of these modified leaves is particularly thick and strong, making these, pointy and sharp and durable. So spines have a reduced surface area to reduce water loss and a thick cuticle that discourages herbivores. Another type of modified leaf is something called an insectivorous leaf. So yeah, insectivore means eating insects, and it's exactly what you're probably thinking of. It's the Venus flytrap. So this is a modified leaf that can that has touch-sensitive trigger hairs on the inside. A fly or some other sort of insect comes in, it clamps shut, and then it digests this fly. This is not the only way to be insectivorous, this sort of trap mechanism. There are also insectivorous leaves that are like pitchers. These are pitcher plant style modified leaves where they form this watertight pitcher that's filled with digestive juices. Bugs climb in, they can't climb out, they get stuck in this fluid and they are digested. And there's a third type of insectivorous leaf that is like a sticky trap, where flies or other insects land on these things. They get stuck by these secreted compounds, and they're just stuck and they are digested. Now, what you might be wondering is, doesn't this count as heterotrophy? Something we said at the very beginning of this plant, all these plant chapters, was that plants are autotrophs. They're supposed to be doing photosynthesis. They're not supposed to be eating stuff. Well, because of a technicality, this is not technically heterotrophy. Heterotrophy means you're eating stuff to get carbon. The reason why these insectivorous leaves evolved was not to get carbon, it was to get nitrogen. These plants live in very nitrogen poor soil, and you need nitrogen to live. Everything needs nitrogen to live. So what are you going to do if you're a plant and you don't have enough nitrogen? Well, you know what has lots of nitrogen? Meat, or protein technically, has lots of nitrogen. So by capturing these insects and digesting them, you can get lots of nitrogen to supplement, what the soil doesn't really have for you. So these insectivorous leaves trap insects specifically to supplement nitrogen uptake. Okay. There's one more section in this plant form and physiology chapter, but I don't think I can squeeze it in time. So I think this is where I'm going to end lecture 2.5. We're not done with this chapter, but the last section of plants is going to be part of exam number 3, not exam number 2. This is the end of 2.5 and our cutoff for exam 2.